So good morning, everyone. So welcome to the keynote address today at SMI conference. And it's our great pleasure today to have Dr. Vince Calhoun as our keynote speaker. So before we start, I give an introduction for Vince. Okay, so Vince is the co-founder of the Tri-Institutional Center for Translational Research in Neuroimaging and Data Science, we call TRANS, and Georgia Research Alliance eminent scholar in brain health and image analysis. So Vince has started the TRANS Center, I think, a couple of years ago when he moved to Georgia, and the TRANS Center has done wonderful, wonderful things in brain science. Vince holds appointments at Tri-Institute at Georgia State University, Georgia Institute of Technology, Georgia Tech, and Emory University. He was previously the president of the Mind Research Network and distinguished professor at the University of New Mexico. So Vince always amazed me in terms of his productivity. He is the author of more than 850 full journal articles and over 850 technical reports, abstracts, and conference proceedings. So his work includes the development of methods to analyze functional neuroimaging data, such as independent component analysis, deep learning for neuroimaging, data fusion of multimodal imaging and genetics data, neuroinformatics tools, and the identification of biomarkers for disease. And he's an expert in studying schizophrenia disease, as I know. So Vince is a fellow of numerous institutions, include the IEEE, the American Association for the Advancement of Science, the International Society of Magnetic Resonance in Medicine, and American Institute of Biomedical and Medical Engineers. So he served as the chair for the OHBM, or Organization for Human Brain Mapping, from 2018 to 2019. And is a past chair of the IEEE Machine Learning for Signal Processing Technical Committee. Vince currently serves on the IEEE BISP Technical Committee and is also a member of the IEEE Data Science Initiative Steering Committee, as well as the IEEE Brain Technical Committee. So without further ado, let's welcome Vince and to deliver his keynote speech. Again, we're very happy to have you here, Vince. And last thing I just want to add, Vince has presented in statistical conferences actually on various occasions. So we're always happy to have you to present your work here. Okay.
Thanks very much. So what I want to do in this talk is try to kind of give a survey of looking at whole brain connectivity dynamics, primarily in functional magnetic resonance imaging. And so kind of survey a bunch of different approaches and kind of go through a little bit of a history. But let me just start first by saying, this field of sort of whole brain connectivity is started with a few papers in, it's fairly recent, about 10 years old or so. Initial papers from Katie Chang and Unal Sakaglu, basically looking at kind of sliding window approaches to functional connectivity in fMRI data. Followed by Visa Kivanimi's study in 2011 looking at voxel-wise changes in default mode network. Things started to really take off, I think, in the subsequent years, 2012 to 2014, with a number of papers looking at connectivity states, dynamic graphs, functional network connectivity states in multiple patient groups. And there's quite a few reviews that have been out there. This one from Matt Hutchison is a pretty comprehensive one from early on in 2013. I have used the omic notation again in 2014, defining a term called connectome. And this one from Dan Lurie is, I think, a good one to look at if you just want to learn the field a little bit. It covers a lot of issues, including methods and application, validation, questions about what is this fluctuation that we're looking at, and how do we know it's real. And then Armin Iraji's paper looking at spatial dynamics, which is, I'll get into kind of temporal and spatial dynamics in a bit. So there's been kind of a lot, and that's just a real sort of brief survey. Things have really started to kind of take off. And so what do I mean by this? So in resting fMRI, and as you probably know, we're looking at sort of coherent fluctuations over time when we're looking at functional connectivity. We primarily focus, and most of the slides I'll talk about today, use sort of adaptive data-driven atlases as our sort of initial step. So we're using independent component analysis here in a lot of cases, where we basically extract, maximally spatially independent components, which I'll call networks as well. Each one of those has a temporal signature to it, and so what we can do for each of these is take their temporal signature within each subject and compute the covariance within subject. And if we order these in a way, sort of anatomically based, we see this nice modular structure in this network, which I'm going to call functional network connectivity matrix. If we move to dynamic FNC or time varying FNC, a simple way to do that is to just use a sliding window where you compute the covariance as a function of time now. And then following that, you can do a whole bunch of different things. You can, for example, take these and just perform a simple clustering into what I will call states, which are essentially connectivity patterns that an individual moves closer to or farther away from over time. You can use, Markov modeling and estimates, which state they're closest to or which state they're in at any given time. So this subject started in state 3, then 2, then 4, et cetera. And essentially, we're trying to get at, looking at this data in a way that respects the fact that we know everybody's doing different things at different times. The brain's a highly dynamic organ. And especially if you're looking at resting fMRI data, it's a very unconstrained type of a paradigm. So one sort of comment before I jump into more details on the dynamics. I think it's really important if you're studying dynamics especially, but connectivity more generally, to use adaptive atlases, because if we have a region of interest, right, that we've kind of fixed from anatomy, for example, it may not reflect coherence within an individual subject. And so we might be making, incorrect assumptions about the results if you've got sort of different populations contained within different regions. So one of the challenges, though, with this sort of data-driven approaches like ICA is that if you run it on an individual subject or even on different groups, you might get slightly different. You have a comparability problem across those analyses. They're not exactly the same. So we've kind of started to do for these sort of scaling up and looking at large amounts of data, we're using these kind of hybrid approaches where we take component maps that we've extracted from multiple large studies here. So we have 2 studies here. We did separate ICAs on those studies, and we found component maps that replicated across the 2. And then we fixed those and use those as spatial priors for a spatially constrained ICA. And so then what we do is we go to individual subjects and we estimate, maps and time courses using that sort of spatially constrained algorithm. And so the advantages, I think, of this type of approach are pretty clear. If you look at the right here, you can see 2 independent data sets, the functional network connectivity, very, very highly similar across the 2. So it's sort of robust and replicable. But in addition, if we look at these subtle group differences here, looking at schizophrenia patients versus healthy controls, you can see how similar those left and right matrices look. Again, 2 completely independent data sets. Thirdly, since we're estimating on individual subjects, it makes cross-validation a lot easier because we don't have to worry about leakage between subjects, for example. And we've kind of compared, as I had mentioned earlier, that, looking at sort of these whole brain networks tend to outperform looking at fixed regions of interest. And these fixed ROIs can again, mislead you in some cases. Okay, so back to dynamics. So in one of our earlier studies, this is just to kind of show an example of states here, estimating from resting fMRI data. So this was, a few hundred subjects, and we're looking at states for schizophrenia patients and healthy controls. So there's 5 states here. So a couple things you can do with this, or a few things you can do with this, is we can look at cell-wise differences in these states. And so one of the things we noticed early on was that the differences we see in the patients and controls are kind of split up in what appear to be meaningful ways across these different states. So for example, here you see this sort of thalamic, subcortical and sensory motor strip here showing a relationship. And there are different patterns in these different states. Secondly, we can look more at the behavior over time and quantifying that. So things like the dwell time, how long do individuals spend in each state? And we see a significantly more time spent by healthy controls in states 1 and 2, which are more modularized states. Whereas these sort of weaker states 4 and 5, schizophrenia patients spend much more of their time there. And we can also get at more explicitly dynamic measures. One example here is what we call a metastate approach. So this is really looking kind of at a state, a state modeling type framework where we have state 1 versus state 2, and there are levels, sort of the degree to which an individual is either participating or anti-participating in each state. At each time, and you can see this subject kind of jumps around in this way, and here's another subject at 4 different time points. And we can quantify the behavior or the trajectory here in these vectors, which we call metastates. And so this is the first state, this is the second state, and the value of the vector tells you the level that they're in. So is it strongly positive, strongly negative, etc. And so then once we create these sets of metastates, we can quantify them in a lot of interesting ways. We can just count the number of unique states. We can look at the number of changes. We can look at the total distance traveled, how far out they went, etc. One of the things that kind of convinced me this was a meaningful approach, because this is kind of a global approach. It's kind of summarizing across all the states at once, is that regardless of how we estimate these states, we can use, I mentioned clustering, but you can do PCA, you can do temporal or spatial ICA, you can do other things. When we looked at, in this particular data set, schizophrenia versus healthy controls, we see the directions are all similar in that we get negative, we get sort of overall reduced dynamics, reduced dynamic range, regardless of how we estimate the states. And so the states that come out of these 4 different approaches are different. They do not look the same because we're optimizing for different criteria, obviously. And that's fine, but it's also nice to say that, well, if we kind of combine it all together, we get similar answers in this case. Another area that's kind of growing, I think, in interest is, well, how do we link together activity, which is sort of one time course in one region, and connectivity, which is pairwise relationships between the two. And so we can look at dynamics in both of those and look at the links between them. And a simple way to do that in terms of activity would be to look at something like the amplitude of low frequency fluctuation, or ALFF. And so essentially we compute the spectra, and then we take the normal, we normalize it, and then we compute the power under a low frequency, particular frequency range. And that's called ALFF. There's a bunch of studies out there on this. But we can do this in a windowed way as well, and essentially get variations, fluctuations in ALFF, do a similar type of clustering. And so we get these ALFF clusters here, you can see. And so I'm showing you kind of as a vector, but also mapped back into brain space down here. And then we can look at how those are linked to the functional network connectivity states. I'm showing you 3 different states here. And these are the correlations between the ALFF and the FNC. And notice how strikingly different across the different states they are. Also, it's very, there's a strong interaction between schizophrenia and healthy controls in diagnosis. So you see, for example, quite different behavior in the linkage between the two of these in the two groups. Getting at information that, again, we're missing if we don't look at both pieces, activity and connectivity. We can also look at things like local efficiency. So basically characterize the graph properties of the states and look at how that correlates to the ALFF metrics in their different domains here. So we have subcortical, auditory, sensor motor, et cetera. And we're seeing, again, some really interesting relationships where we get these sort of positive correlations in some states, negative correlations in others. Okay, so temporal dynamics, I'll kind of continue in kind of what I'm talking about, or what I've been talking about, and just show a couple different ways to look at this. So you can, for example, again, all the things you can do with temporal analysis, you can apply to resting fMRI data in this case. So here's a time frequency analysis where we used a wavelet transform approach. And so essentially what we're doing is jointly estimating the amplitude of each state, its coherence, is it anti-correlated or strongly correlated? That would be the color. and its amplitude. Is it white or low amplitude, or very strongly intensely colored, which is a high amplitude? And if we do this, we see some really interesting things where we can start to separate states that have similar amplitudes but different spectra, and vice versa. And we see that if we do that, we actually can capture meaningful relationships in the data that are linked to certain variables of interest, whereas other ones are not. And if you kind of have them sort of, if you don't look at it where you separate out based on these different measures, they get mixed together and blurred out. Another example is to look kind of more naturally at the trajectory. This is work from Ashkan Faghiri, who's a graduate student in my group, and he's basically looking at, you know, the full trajectory over time, essentially looking for sort of density based measures. So he has this approach that he's using based on energy, which is really looking at kind of clustering and then representing the states that, you know, what's happening at these different points in time. Something that comes up a lot as you do this work is, obviously, if you start with kind of just a very simple sliding window approach, it's, well, you have to pick the window and there's averaging that occurs, and that's certainly true. So for example, here, if I have only 2 states in my data, but I use a sliding window, I'm probably going to estimate that there's 3, because I have one that represents the overlap between the 2. So it could be 3 or more, for example. And you can relax the window assumption, which is essentially a filtering based assumption. And so we have an approach that we proposed, which is based on dictionary learning, where we show that, you know, a sliding window approach doesn't represent the ground truth as well if we have things that are kind of randomly shuffled and they're transitioning very fast, et cetera. The good news is that, you know, in this type of an approach, what we found was that the patterns that we observed that came out in this windowless approach were pretty similar to what we found in the sliding window approach, with the exception of one that tended to be kind of weakly correlated in sliding windows. But we had these strong patterns that we found when we looked at the windowless approach, suggesting, and actually it was true when we quantified it, that this state was much more transient, kind of in and out. So the window was sort of blurring it. Ashkan actually has extended, you know, this concept a little bit to say, well, let's, instead of modeling this as, you know, just we pick a window and just do some analysis, let's be a little more systematic about it and use a system where we have 2 different time courses here, and then we have a set of filters that we're applying, and then we have a filter bank at the second level where the connectivity relationships are being estimated. And this lets us look, you can set those filters to have different properties, but in this case, we're looking just from low to high. So across all the frequency bands that are available to us. And, you know, obviously it's pretty simple in that it works well. If we have a slowly varying, you know, connectivity time series, we can capture it with a windowed approach. We capture it in the low frequency filter bank approach. If we have a rapidly varying connectivity, it gets missed by the sliding window, and it gets well estimated by the second band of the filter. This gets really interesting when we look at actual data here, and we see, for example, the states that are estimated. And in addition, you can see the different filter bank properties. Again, this is frequency from low to high. So again, we get some that are dominant low frequency, others that are more broad band, and others that are dominant high frequency. And we also see highly significant group differences in the relationships to these different bands. And then the last one I'll mention is just instead of looking at just the connectivity over time, sort of, you know, this sort of movie, for example, and clustering that movie, we can look at we can start to get a little bit more detailed and look at its first derivative, right? So now we're looking at change of these connectivity patterns, change of change. And if we do this, and then basically just, again, fairly straightforward, we're just taking the changes over time and their first temporal derivative, performing clustering in that space. And what we find is that, you know, in some cases, we see some states that have different clusters that have very similar amplitudes, but opposite signed derivatives, right? So those are essentially pairs of clusters that when someone gets into cluster 1, it's much more probable that they'll transition to cluster 2. And so they tend to oscillate back and forth between cluster 1 and cluster 2. Same for state B. And so you can think of those as kind of an attractor in a sense. And so we've modeled it that way. And then we looked at it in the context of mild traumatic brain injury. And this is actually some of that data. And what we found was that if we look at state A and state B here, we see that in this longitudinal data set, after injury, we see a disruption of the link between these two state attractors. After recovery of 4 weeks, we see a partial recovery of state A, and then 3 to 4 months after injury, we see full recovery in the data. Okay, so now let me just talk about spatial dynamics, and what do I mean by that, and what's the terminology I'm using here? And so I think it's just pretty clear from this picture what I mean. So temporal dynamic is essentially fixed nodes, and then you're looking at changes in how they couple over time. So the correlation in their time series, for example, how that changes over time. Spatial dynamic would be, well, let's let the nodes change. Maybe they shift a little bit. Maybe they shrink and grow. Say shrink and grow. And then spatial temporal dynamic would be allowing the nodes to change and the coupling between the nodes to change. Okay, so those are sort of sequentially different models there. And here is just a couple examples to show, that we think this is really important. And so here I'm showing you examples from 3 different domains. Let's just focus here in the middle on this subcortical domain. And we basically modeled it with a set of pretty fine-grained spatial basis sets. And we see that the patterns are coupling together differently over time within that domain. And we see, for example, if we look at the spatial dynamics alone in the context of schizophrenia, we see, thalamic dynamics, for example, are reduced in interesting ways. So this is really just looking at a hierarchy, right? So it kind of pushes the problem back a little bit to kind of a more fine-grained and more large-grained level. But we can also look more directly at sort of voxel-wise dynamics, or essentially then we can start to look at fluids and things like that, fluid models and the like. Just to kind of highlight one result, if you look down here at the default mode network, we're essentially estimating default mode network variation over time at the voxel-wise level, and then estimating spatial clusters from the data. And we have 3 different spatial states. And what we find is, for example, certain parts of the default mode are always in all 3 states. Other parts are sort of uniquely and transiently in only one of the states. And this gets at some, it kind of touches on some of the unique work that Randy Buckner's group was doing, looking at fractionation of the default mode and how it's different in different people. We're kind of pushing that a little further and saying, well, it's actually different probably because these individuals are spending more time in these different spatial states. And so it really depends on how you estimate from that data. And so, I think that's kind of a really interesting direction to pursue. There's a lot of work to do here still. If we start to look at spatial patterns, though, we have to come up with different metrics. So, for example, one that we can do is use essentially a spatial histogram and based approach, or sort of and look at how those vary, for example, in patients versus controls. Do the distributions of these maps tend to shift spatially from one value to another? And this table is simple and simplistic in some ways, but we're trying to kind of make the point here that a lot of the work out there doesn't differentiate between whether it's temporal, spatial, or both. Most of the work that has been previously done, the dominant amount of work is temporal, looking at temporal dynamics with fixed regions of interest. Any method can be extended to do this, but a lot of the work hasn't. And so I think as a field, we need to be more clear in terms of what these methods are doing and what are the modeling assumptions underlying it, because I think that will matter to us as we go forward. Okay, so next I'm going to talk sort of about these classification, simulation, validation concepts. One of the things that we found early on was that if we look, and it makes sense to me, if we look at the data dynamically, we know it's highly dynamic. We know it's unstructured, in terms of the paradigm. We actually can improve our ability to classify patient groups. And so this was looking at a 3-way classification of bipolar, schizophrenia, and healthy controls. And we directly compared the use of a static connectivity matrix to functional connectivity matrices. And these are all cross-validated, and there's a bunch of steps in here I'm kind of glossing over. But essentially what the take-home message that we found was that static does well. So about a 60% accuracy in a three-way classification, so the chance is 33. The dynamic approach was substantially higher at about 84%, and combined was slightly higher than that. And we see this when we do simulations. We also see this when we look at sort of cross-validated classification accuracy. We get the biggest bump from going from one state to two, right? So you can see these are again all cross-validated, but we go from 1 to 2, and the accuracy jumps up. And then there's some variation depending on how many states you're looking at, in those individuals. We see a little, in this particular data set, there was kind of a maximum accuracy around 4. So that's sort of getting at, potentially using this as a way to estimate a model order. Selecting the number of states is another model order problem, right? So again, lots of unsolved issues, lots of methods work to do there. We have looked a little bit at a variety of approaches to do that. We looked at, in this case, 24 different indices, including things like the elbow criteria. At least the work that we did, we converged on this Davies-Bolden approach and Ray-Turi approach, which tended to be the most accurate relative to the ground truth measures that we had in that paper. And then for those of us that are trying to sort of validate these approaches and do simulations, et cetera, just to kind of note that it's really, you have to be really careful in how you do this and what is your null model. Obviously, if you say, well, you're looking at dynamics and your estimate is actually more of a static measure, then that's probably not getting directly at the question you're trying to answer. Also, another note, which again is hopefully probably obvious to this audience, but the term stationary has a very clear definition, but it's really an ensemble level approach. And just because something is stationary by definition doesn't mean there aren't lots of interesting dynamics occurring, locally within that data. And so, we kind of simulated 4 different categories here, including, stationary with no epochs, and stationary with Gaussians, real fMRI data, and then explicitly non-stationary. And then we were trying to come up with sort of, dynamic detectors in a sense, right? And so there, if you look at something like kurtosis is really good at detecting outliers, right? It's good at picking up motion. It's not very good at picking up interesting dynamics. We used a wavelet-based approach called multivariate phi, which appears promising and sort of reflected what we saw in our simulated data accurately, although it's still a work in progress, and I think there's a lot of work still to do here. We've also looked at replicability in real data. So this is looking at about 7,000 data sets. We split them into 28 sets, did separate estimates, separate dynamic quantifications, and looked at their differences in their metastate metrics, and you can see the variation here. If we disrupt the dynamics, using some sort of phase randomization, for example, pretty clearly breaks the models as we would have expected. Importantly, we found pretty high replicability. We found evidence that these states, suggest that they're somewhat canonical when we looked at these large data sets, because we saw them, similar states occurring across these different independent sets. We can also look at concurrent EEG fMRI to again get at more what is the fMRI telling us. And so with EEG, I think, as you know, it sounded like from the previous session, you can estimate measures of drowsiness and sleep, and you can link to kind of well-known rhythms like alpha, theta, and delta. And so taking the EEG data and then looking at the relationship of those to fMRI in terms of drowsiness, we see that the fMRI state most linked to EEG measures of drowsiness had, of sort of alertness, had the most anti-correlated states, where we kind of shifted to being less anti-correlated and then almost to a hypersynchronization when it got into the more drowsy based points in time. We can also do predictions. We can classify sleep and wakefulness from from fMRI data, from fMRI states. And this was again done with the concurrent EEG fMRI measure using some data from Enzo Tagliazucci's group. And so we were able to basically accurately predict the sleep hypnogram from computed from the EEG measures. But interestingly, that's not the only story. If we can also get replicable states within just the wakeful condition that are also predictable, right? So I think there's a lot of work here still to understand, what can we see and what's meaningful in this fMRI data and in these fluctuations. And then the last one here on the sort of, what is this telling us? And, is sort of looking at tasks, right? Because we can modulate patterns by having people do explicit tasks. And so this is a real sort of brute force task. Javier Gonzalez Castillo developed this. And so they're just looking at blocks of rest or memorization or video or math. It's pretty intense. And that's reflected in the dynamics as well, in that if you just estimate, since we know a priori there's 4 tasks, if you estimate, or 5, and if you estimate states in that model, 4, right? If you estimate states in that model, you get almost exactly back patterns that reflect that, right? So as you might expect. But then if you go another layer deep and look within just these periods of task, you can start to see states occurring within the tasks as well. And so I think, it is important to recognize that sort of the amplitudes of these things are a little bit different, in that task is a pretty strong measure it influences connectivity patterns fairly strongly. Arousal does as well. And then underneath that are probably other interesting sort of cognitive-based fluctuations, cognitive-driven fluctuations, and others. Okay, and so this last one is just looking at going even a little bit further in our modeling to try to, again, unpack the dynamics in these data, kind of searching through the dynamic space a little bit. And so I'm just kind of highlighting some high-level approaches that we've used, one of which is sort of an approach we're calling tri-clustering, sort of builds on bi-clustering. Essentially what we're doing is identifying patterns that have similar values, cell-wise connectivity values, similar change over time, and subjects that show similar patterns. So we're kind of jointly clustering on all 3 of those measures. And so here are some examples of those tri-clusters. And what you can see in the notation down here is that we have in this tri-cluster 3,500 windows, 81 subjects, 619 cells or pairs, right? And so these are other examples of that. And interestingly, when we do this, we actually increase our sensitivity to, for example, group differences. We also increase our sensitivity to other variables of interest, presumably because we're comparing more apples to apples as opposed to looking at the entire fluctuating data set and trying to sort of test in that big set of things that varies quite a lot and has a lot of different inconsistencies. This is some work from Apo Hyverinen's group looking at essentially temporal primitives, estimating temporal primitives using a deep learning model. And what they found was you get these replicable sort of chirps almost. Well, this one's a chirp, this one's more of just a blip. This is an anti-blip, for example, that were linked to, they looked at resting data, task data, as well as behavioral traits. So again, I'm not really trying to summarize, these studies in depth, just giving a little teaser of some of the ways we're trying to unpack this complex data. Another example in the primitive type direction is what we call statelets, where so you may have heard of approaches that are focused on looking at recurring shapes in time series. So that would be called shapelets, right? So we're essentially kind of taking that concept, but doing it in the context of these pairs, so these matrices, and looking for signatures of these cells that recur over time within subjects and across subjects. And so we basically start with the same time series, then we do this sort of exhaustive search for what we call motifs. We can look at how those motifs cluster in a 2-dimensional space here to see if they're kind of separated from one another. This gives us an example of relation, the sort of max connectivity strength within each pair looks like, and how the differences, this is a difference between patients and controls. And you can see some are very strongly positive, some are very strongly negative. But the most interesting sort of take home from this for me was the transit time results. So if we look at these motifs and then we look at how long they are, right? And we look at that by group, we see very striking differences in that healthy controls tend to have much longer transit times. It's consistent with the dynamic, reduced dynamism we saw before, but schizophrenia patient transit types are much shorter. So you can see here they start and they stop very quickly, whereas the healthy controls persist. This is just one subject. This is a distribution across an entire data set. You can see very, very different. And then again, if we didn't have enough to look at already, we're trying to move here to let's take these networks and try to get at spatial temporal dynamics, right? So we basically use ICA networks templates as priors, and then use a deep residual network model developed by Benim Kazimavesh as a graduate student of mine, and basically for each network we're estimating a spatial temporal network. Basically, we're expanding that network to look at fluctuations in both voxels and time series. And so then we have now a 5-dimensional data set. So we have space, time, and network. And so once we do this, we've now, in some ways, it's nice because we separate the data into fluctuations that are linked to different networks. Then we can use metrics to summarize and reduce that back down to something we can kind of understand, has some intuition to it. So here, for example, if we just look at with the, one of the motor networks, if we just look at the average over time in 2 different subjects, you can see there's variation across subjects. We can compare clusters, spatial clusters within that same network, and see if it's related to, in this case, we were looking at age, and we see some of the clusters are strongly related to age, others less so, some not at all. And then if we look at fluctuation voxel-wise, in this case, we're just looking at variability, so it's a pretty simple measure. The left side here is the amplitude, kind of what I showed you up here, but it's across subjects. We see a very, we see the sensory motor network, but we're missing that, the sensory motor strip, the rest of the strip. If we look at variability, we see these really interesting focal spots of high fluctuation voxel-wise that go along this strip that are completely missed by this amplitude approach here, suggesting that this is complementary and useful information. And we can also, again, look at, if we look at the time series here, we did some reduction again, where we kind of average within voxels that are strongly in the network, and we look at changes over time in the functional network connectivity. Again, lots of things you can do here. It's really just getting at, let's try to put the data into a space that we can start to use and help interpret. And then the last one here is looking at some work from Robin Miller, who's here at Trends. And so she's really trying to expand the concept of these states, these dynamic functional network connectivity states, which she calls snapshot dFNCs. So essentially they're just sort of, again, we're estimating clusters, right? So they're just sort of 5, in that case, fixed states. But expanding that to say, well, maybe they evolve, maybe they fluctuate up and down in different meaningful ways. So she's kind of dubbed that EVO dFNC, or multi-frame evolving dFNC. And so just at a real high level, and I'll provide, I think we're going to put this up on bioRxiv soon, so you can take a look at the methods, but we're essentially using a UMAP embedding approach with a bunch of little tricks, basically constructing a linearized 2-dimensional dFNC trajectory that we can then, once we get this space, we can look at how that trajectory evolves over time. So this is a movie here of FNC for one state. So it's basically a movie state. Why would we do this? Well, we started to look again at the relationship to patient control differences. And so if we just look at exemplars sort of clustered across this mapping, so in this case, we have 10 of them, 10 different movies here, and we characterize the differences in schizophrenia patients and controls. We find very strong and striking differences in both directions in these different movies. We find sets of these movies are strongly related to positive symptoms, which are notoriously difficult and sort of in some ways hard to link to imaging data for lots of reasons. But it appears that in this sort of movie state space, that does appear to be an easier representation for mapping to symptoms. Okay, and I think I've got a little bit of time. I've got about maybe 4 or 5 more slides just looking at some different clinical applications, just to give you an example. There's been so much work out there, and I'm highlighting work from our group primarily, but many others, lots of other groups doing lots of interesting things in this space. And a lot of it has been applied to clinical data already, and for good reason. We can see, for example, that if we look at things like mood and psychosis, so schizophrenia, bipolar disorder, schizoaffective disorder, we see that there do appear to be differences in the way the states are structured across these different groups. We also see, in this case, looking at ADHD, multi-scale effects. So for example, we could look globally at the entire brain and look at the dynamics at that entire brain. Or we could say, well, I just want to focus on the default network, right? So I've got maybe a set of 5 or 6 or 10 nodes within the default mode network, and let's look at the dynamics within that particular network. And we get basically opposite effects at these different scales, right? So the directionality has changed. So I think it's getting at this as a complex problem, and obviously looking at things at different scales gives you different answers for different reasons, right? So there's still a lot of work here to do to figure out what is the right way to look at this, and I actually don't think there is one right way to look at this. I think it's important just to be very careful, systematic, and come at it with a particular question of interest. The dynamics have also looked to be useful to separate subgroups in data. So here's an example looking at subcortical ischemic vascular dementia, which is sort of a subtype, a vascular subtype of AD, so to speak. And so we're seeing, basically that the states are significantly separating these different groups. For example, the Alzheimer's patients had more transitions between state 2 and 3 in both directions compared with the SIVD patients. And again, substance use, is in some ways it's comorbid with with a lot of studies of patient data. So it's really interesting to start to try to look at that. Also, just to look at substance use in and of itself. Here we were looking at the dynamics related to multiple substances and their combinations. So basically alcohol, smoking, marijuana, and then different combinations of those. It's not a huge data set, but it was enough to do something we thought meaningful. We asked the question, do these states kind of differentially align, and do they show different sensitivity to these different combinations of substances? And indeed they do. For example, state 5, was showing a strong relationship to, essentially most of the substances here, right? State 1 was kind of specific to smoking, for example. And I think, again, as I had mentioned early on, prediction is going to be, sort of sensitive predictions from fMRI data is going to essentially need to use or leverage from dynamic approaches. At least I think it kind of cleans up the data a little bit. And so here we just compared in mild TBI the AUC for looking at either diffusion data, resting fMRI data, or dynamic resting fMRI data. And again, this is a small study, so I don't want to make too much of a deal out of it. But we also are looking at the impact of pipelines and movement and things like that on static versus dynamics. And it appears that we can start to separate out some of those artifacts a little better as well if we look at a more dynamic analysis. And this is the last slide. I just kind of wanted to highlight, or sort of the last results slide, just to highlight, or I could give a whole other talk on sort of data fusion between multimodal data and dynamic measures, but this is just one sort of little teaser from a couple years ago. Barnley Rashid did this work, and we asked the question, are single nucleotide polymorphisms genomic data, essentially patterns of genomic data, associated with these dynamics? And the way we did this was we computed a polygenic risk score for schizophrenia, and then we looked at its relationship to the states that were estimated, the same ones I mentioned earlier in the talk. And so what we found was that this state 1 showed a strong relationship to polygenic risk score. It was a negative relationship, and so the more occupied this state was over time, the lower your risk for schizophrenia. And so if you remember, that state was also the one that we showed that healthy controls spent more time in state 1. So it's consistent, and so the genetic risk score is supportive of that same fMRI result. So I think that was, for me, a pretty interesting result. There's obviously, again, a lot more to do here. That's kind of my theme throughout, I guess. We do try to release our software. I think virtually all of the methods I mentioned, and soon all of them, are released in either our GIFs toolbox or our FIT toolbox. It's a MATLAB toolbox, but we also release executable versions, and we have a Python binding for it. We also have a containerized version of it, so you can use it in various tools these days. If you're interested in learning more about dynamic connectivity, I'd recommend this review from Armin Iraji. We don't just cover the work that we do. We try to do a broad survey of the field a little bit. And we also introduce the dynamic FNC toolbox that we've built that's a subset of the GIF tools. And this is just kind of some examples of the kinds of output that you get from that. And with that, I just want to also thank everyone at Trends who have done most of this work. And also thank our funders and many collaborators, and also to you for bearing with me and hanging in there throughout the talk. At least I think you are, because I can't see if anybody's still here. So with that, I will close.